Hi, my name is Alex Moreau, and my topic is the rise of limbs from ancient fish, or the origin of the limb from the fin. So first, just a funny meme I found. I thought it's really relevant. Um, so first, before even talking about Tiktaalik, just quickly, so when someone says transitional species, what they really mean is transitional fossils. There's really no known examples of living transitional species. More often than not, it's fossils. Um, and what classifies a transitional fossil are animals or species that have traits from both their ancestral group and their derived descendant group. Um, so speaking about Tiktaalik rosae specifically, it was first discovered in 2004 by Edward B. Deichler in northern Canada. Um, Tiktaalik had features that were common both in the primitive fish and early tetrapods. And I mean, we saw a picture of what Tiktaalik looked like in class. It looked very amphibian-like. Um, Aside from those traits, what made it really remarkable was its ability to stand on land and the unique anatomical structure of its fins. It allowed itself to support itself in a standing position, but based on the papers that I've read, it it's very likely that it had very limited range and motion with its fins. That it probably couldn't walk out of water very far. Um, but once it was out of water, it was able to support itself because it had a reinforced spine, which would allow it to withstand the added weight of being out of water. So just some of the things I talked about before, some of the anatomical evidence. And there's another species that I looked at as part of the study for my paper, and it was Rhizodius herberti, which is more fish-like than amphibian-like, though it had features of tetrapods in its anatomy. So it's also considered a transitional fossils. Um, what is interesting is the researchers in the paper I read found evidence that the bones of this species were actually formed from bifurcating chondrogenic condensation of the different anatomical regions of primitive fish to form like the femur, um, the radius, the ulna. Is very interesting to just read about how the different anatomical features of the fin kind of just coalesced to form different bones. Um, one interesting thing to know is that the Australian lungfish Neoceratodus has evidence of the same type of condensation in its pectoral fin. Um, there hasn't been anything else seen that has this type of evidence, but the authors note that this is most likely due to the fact that this is really the only species that's been studied in depth for this. Uh, and that's probably why it's only been seen in that particular species. So, in addition to anatomical evidence, there's also genetic evidence. Hox genes, just real basic. They're we all learned in genetics. They're one of the genes that are responsible for body planning during the development of the embryo. Uh, they in the study I read, they were looking at zebrafish. A lot of studies with zebrafish, and they actually were able to. So they actually overexpressed specifically Hox D13, and when that particular gene is overexpressed, it actually caused the zebrafish to um, generate some additional endoskeletal elements. The authors surmised that this would mirror morphological alterations that were incredibly crucial for the development of the limb. So it's very similar. So it's very possible that what the researchers saw was the very beginning of limb development. Another thing to note that I thought was interesting is that the researchers found that any change in length, either longer or shorter, of the amino acid repeats, seemed to correlate to phenotypic variation. And there's actually an example of this in the modern world. The dog breed Great Pyrenees, I don't know if I pronounced that right, actually is the result of variation in amino acid length. Specifically, it's the shortening of the of glutamine repeats in ALX4 and bilateral polydactyl development, which creates that breed. Variation in length also occurs much more frequently than point mutations do. So mutations based on amino acid variation, occurs more than point mutations. And the last study I looked at chick and mouse studies, and it was found that Hox genes play even more of a pivotal role in those studies. They also looked at GIL3 and SHH. They found that GIL3 repression could have resulted in fin movement on the anterior side of the skeleton for ancient fish. And SHH expression could also lead to repression of anterior limbs. So like getting rid of the fin and, maybe pushing things towards the other side. And that's it. Thank you.